It is with really a great honor to present our keynote speaker, Ned Mozier, as our opening speaker. Ned, if you want to come up to the table. So Ned has been in the industry for 35 years and recently retired as a VP of CMC development from Pfizer. You know, if I had to count the number of products that Ned's worked on in commercializing, from biologics to vaccines, it would take all day, right? He's got a tremendous wealth of experience in doing this. And one of the things that I've known about Ned is that he is a stalwart scientist, a champion of driving scientific innovations in solving complicated CMC solutions. And so it's a pleasure to have him, and welcome.
Thank you, Bhavin, for that kind introduction. And thank all of you for attending this meeting. I can't tell you how great it is to be in a room full of scientists and like-minded folk. After a year of retirement, been out with a lot of different people. And it just feels like coming home again, especially with bioassay folks, near and dear to my heart. A job I held for a decade or so within Pfizer. I did have a different job for the last 7 years before I retired, so my information and technology is a little bit out of date. So what I'd like to talk about with you today, to just kind of kick this conference off, is my view from a little distance. And most of this is what I remember. I do have some information, some data, but it's not like when I was at Pfizer and I was swimming in a world of data and I could pick and choose anything I wanted to. So it's a different kind of talk for me. I'll try to make it interesting and relevant, and hopefully it'll stimulate your thinking and kick off this conference, which has some tremendous talks that I'm really looking forward to. I use the word special because it's so unique what we do in a bioassay, and it's so difficult to explain it to folks who aren't familiar with it. So I'll talk quite a bit about how to communicate what we do to other folks, other scientists, as well as the public. If you boil down my abstract, one way to explain what we do is it's typically the only test on a spec list that can detect a meaningful conformational change. So it could be the way it's made, or it could be the way it changes over time upon its intended storage. So that's something that's unique. That's one of the things that makes bioassay special. A little bit more about that. Of course, we all know the downside is there's relatively high variability. It's not the only variable method on a spec list. If you look at an IEF method, there's a lot of noise there as well, for example. But it's such an important assay that when there's variability, it's often confusing on how to interpret and understand the information and what the numbers actually mean. But the great benefit is there's an ability to actually probe a 3D structure of, for example, how an antibody interacts with an antigen or its target binding protein. And I would encourage all of you beginner bioassay folks to think like a molecule. Think about how things come together and interact. That's why I've shown this conformational image. My mom asked me what an antibody looked like one time. And I kind of showed her this picture, and she says, that looks like a blob. And how do you know that? You've never actually seen one. And I said, but we do know. But the bioassay is the only thing that really looks at how these two things interact. So it's a unique test. It is different. Sometimes people think of bioassays as a catch-all. For example, you've got this long list of tests that you run on your product, and there are things, there are unknowns that are going on, and occasionally a bioassay is the only test that will catch that. This was particularly true in the earlier days when the list of test product specs was smaller. And now that we have better tools, particularly for characterization, I would say this happens less often. But it can happen, and it's reassuring to your company to know there's nothing else happening to the molecule that's not detected by these more traditional methods. Make sure you're not missing something. Of course, the question we always get is we run so many tests and we get the same answer every time. You know, it's 100% plus or minus 20% or 30%. And it's very expensive to run a bioassay lab. You know, why are we doing all this testing? We will get challenged, but one of the answers is because it's almost like an insurance plan. You might catch something that no other test did. And we know that the regulators look at this as well. Folks at the FDA have tell me stories about things they have learned, the importance, particularly of the cell-based assays. It's part of the reason they're more popular than the binding assays. Okay, what I want to cover today in my time, and if I could get a 10-minute warning maybe from someone. Thank you. As usual, I've crammed too much information in these slides. I do that. I've been doing that for 30 years and I can't stop myself. I did try something a little different today. We'll see how it works. But I'm going to breeze through the basics and terminology. I want to make just a couple points about that because I imagine almost all of you are very familiar with this. A little bit about my thoughts on designing a method. And I'll spend a fair bit of time on what we call assay noise or variation and how that impacts specification limits because it's so important. And it's so integral to what we do and how we talk about what we do. I'll talk about a few key focus areas, how to use stability study information, and some successful strategies I've learned over the years, lessons learned, key points, and if there's time, which I doubt there will be, I'll talk about correlating assays and bridging assays. But if I don't get to that, all this information is available to you in these slides. Oh, great. I have a timer on me. That's perfect. I need that. Just a few words. I know probably 99% of you know this, but we call most bioassays a relative potency assay. But it's important to be able to explain that to other folks that, you know, it's an in vitro test. It's typically using cells. Sometimes it can be only binding. I say only because, of course, cell requires binding as well. It's always relative to a reference standard, the way it's run. So I'm talking about the more traditional potency assays that we know of. But the word potency is very confusing to folks outside of this room. It can mean many different things. Even within our industry, it can mean the strength of a drug. It can mean the concentration of a drug, a small molecule, or even some biologics, and in some cases, vaccines. So I would be careful not to use the word potency alone, use relative potency assay, and then try to explain to folks what that is. And essentially what we're trying to do in the CMC is not a discovery activity. Essentially, we're trying to establish that each batch is the same or similar enough to those used in the clinical studies. Again, that's obvious to us. It helps to explain this to folks. That's what we're trying to do. It requires a collaboration with discovery because we have to know what the original intent, the intended mechanism of action is. That has to be tied to the assay for us to be successful running a relative potency assay. And also the ability to, under the stability conditions of storage, to monitor a meaningful change and preserve the intended mechanism of action. So, getting started, one of the more important things is to select and choose a well-characterized reference standard. Often it's the drug substance used to make the phase 1 clinical supply. It may be switched once the commercial production is in place and you have your commercial final process. That's not uncommon to do. So here's what I'm trying to do a little differently. I put in very light font in blue. I don't want you to try to read that. That's just kind of my prompt. There's just too many words on my slides, and I apologize for that. So while I was watching the basketball game last night, I did a little editing. Hey, I saw a fist pump. Do we have a Florida fan? All right. How many people watched the game? That was a good one. As most of you know, an ELISA or binding assay is often acceptable at phase 1. A cell-based relative potency is typically expected to support the materials going into a pivotal clinical trial, or a very strong argument with data showing that it's not necessary. And I would say, in my experience, that we have been successful doing that, but you can't just ask for it. You have to come with the bridging data to show that a binding assay is not inferior to use to support material going into your final clinical studies. Of course, you have to lock your cell types in very early because you really need to begin to understand how those cells behave, and it's a lifetime of struggle is being able to get the same cellular performance every day and for the future of these products, and these products will last for decades if we're lucky. And as you well know, if a product has multiple mechanism of action, they all have to be understood and either measured in a quality control setting or as part of a characterization. As Bhavin said, these fracking molecules are getting stranger by the day, and some of them I can count may have 7 activities. You know, if you think of a trifunctional antibody with effector function, for example. There's a tendency to want to do as little as possible early, but you know, it can catch up with you in the end. As much as you can get done early is important because it takes years to really tame a cell-based assay. So the early investment often pays off. You may have to justify this with your management, why you need an organization of this size, but I think that's the communication that you need to have. So it means a lot of testing. By phase 3, you've pretty much invested all the development you need to have, and it's, you know, again, it's an expensive endeavor because even at phase 3, most drugs are not going to become products, even though your probability is much higher. So you're putting a large investment for a shot on goal. Most of you well know what's needed to build a bioassay lab and the particular facilities and the special equipment that's required. I would say the thing I learned is ample hood and bench space. Too much traffic is a problem. I've learned the hard way. You know, you have to advocate to get enough room for your incubators and your analysts. It's a large footprint, if you will. And it's generally pretty boring to look at. When the tourist people come by, you know, maybe you've got a poster up to catch your eye, but otherwise they're just looking at boxes and hoods. And it's not very dramatic. They don't have all the bells and whistles in a mass spec lab, for example. Personnel is important as you're building a lab. You need people with experience in method development, bioassay method development, people with years of experience. And I don't want to underestimate skilled technicians. It seems like, of course you need skilled technicians, but you need people that can run bioassays, and not everyone can run them. You know, I think there's a natural talent that some people have. Everyone can get better, but you know, I could practice every day for the rest of my life and I still couldn't dunk a basketball. But some people can do it immediately. So it's the same with assays. Some people are just very talented in it and some people will never be great at it. and helping those people find the right jobs in your organization. There's always plenty of things to do. For example, maybe someone that's not so great in the laboratory is awesome at looking at the data and trending the data and doing analysis or writing procedures. There's many things to be done in the laboratory. Finding out where the skills of your personnel is very important. You need cell biologists. You need people that understand cells and their behavior, and this isn't something you can learn in a year. This is something that requires education, it requires years of experience. And you need data and software curators. You need people that can run programs, understand how your software programs actually work, adjust them, write them, handle data and trend data in a way that's meaningful and understandable to folks in the lab. And then you need a statistician, at least one. And you need a really good one that really understands bioassays. And I probably went through, in my career, I've probably known 4 or 5. And I would say one of them was not good at all. 3 of them were good enough, and one of them was great. And that great one really made a difference. Do we have statisticians in the audience? Anyone? All right, we got one. So one of the things I say is a design of experiments. It sounds great, and when done right, it's very helpful. But oftentimes they're not properly applied, and people get lost. They don't know what they're doing. They don't understand the principles of DOE. Just a good basic experimental design. And the best statistician we ever had said, I need to understand the science of what you're doing before I can do anything at all. Explain to me what you're trying to accomplish. So that was a learning for me, and I found that to be true. What they really dislike is when you call them in at the end and ask them to wave a magic wand and make your data look pretty and sound better than it is. That is obviously the wrong way to go. Get them involved early. And, you know, we actually had statistician in our group in my last role in Pfizer. Not my bioassay role in Pfizer, and it made a big difference. Of course, you may need people with robotics, liquid handler expertise. And Bhavin mentioned GMP. You know, there's always going to be a question about whether you should run it under GMP and when in development. Starting early, I believe, is helpful. Not everyone can do that. Not everyone has a GMP type lab that they can run at phase 1. So often there's a lot of things that go on outside of GMP, and then it goes to GMP, and it's this massive hill to climb. So be aware of where your bioassay method is going to land and what the commercial laboratory is going to require of you, be it materials or GMP aspects, how you monitor your data. I'll give you just one quick example. You know, we talked yesterday in the workshop, workshops yesterday were outstanding. One of the things is failure rate. You know, you want to fail an assay, you don't want to fail a product. Failing a potential out of spec or out of trend product is a massive undertaking. You want to avoid that at all costs. So you set your assay acceptance criteria up in a way that you will reject an inferior or a subpar, a known performance issue in your assay. Well, one of the things I learned once is that's considered a problem if you're failing 5% of your assays in a commercial environment. It's a problem that has to be solved. And I said, well, no, actually, it's a design feature. It should fail 5% of the time. That's how we design the assay. We want it to, because we don't want to put product at risk. That was a difficult conversation. So be aware of those types of things as you're moving through development. The other point I want to make about the laboratory is pipetting. Everybody can pipette, but if you really watch the way people pipette, they often pipette differently. And there can be a lot of bias coming in how material's drawn up, how it's mixed, how it's released, how much is on the outside of the pipette tips, especially when you're working with small volumes. And again, going back to ability, some people are just a whiz at pipetting, and they do it accurately and perfectly every time. They're just naturally good at that. Other people always struggle with that. So being able to train your personnel to not only run a bioassay, but just the basics of pipetting, for example, is money well spent, in my opinion. It's almost like you need to license your analysts before they go into the lab. Like, you have to pass this test. You have to have a protocol. You have to be good enough, or you're just going to continue to have failures and problems. So I want to spend a few minutes on assay noise, which we all know about, and we get a lot of criticism for, and it's just the nature of cells, and it's the nature of complex assays. So, of course, you need to define the range, the upper and lower asymptotes, and we can argue forever about what specs are acceptable early and what specs are available late. I put a few in here that are fairly common, because I think for the example, I'm going to use them. And early on, your assay capabilities are the driver, because you don't have a lot of lots, you don't have a lot of stability data yet, and as you get further along, your lot release data and your stability data will also factor in, particularly if it's outside of the range of assay noise. Often for monoclonals it's not, but it can be for certain products. So here's a case. I want to explain this data a little bit. Once your method is established and your cell performance is fairly well understood, just testing the product against itself at 100% is a simple and effective development tool, because it really shows the inherent noise in the assay. You're not dealing with sample-to-sample issues. So here's a case where a test is run and relative potency calculated, individual plates, 20 plates over 20 days stretched over some period of time. Here's this data plotted. And you can see right away, if you wanted a spec of 80 to 120%, you're going to have a problem with that. 22%, that seems pretty high, and it is. You want to get to here. So the question is, how do you get there? And again, you could spend years figuring out how to minimize the noise of your assay, or you could take a careful methodic approach and figure it out. It could be the analyst, it could be your cell incubation conditions, the status of your cells, it could be a reagent. Talked about edge effect, particularly for longer assays on the outside. It could be the way the cells are handled. It could be some unknown factor that you haven't put a figure on. This is where the experience of your laboratory comes in, and typically in a bioassay lab, the person developing the assay will seek the opinions of their colleagues, and it helps to have a group to do that, because we need to get better if we're going to be ultimately successful. So how much variability is too much? Again, the same case, testing the same product against itself. You'd like to get a confidence interval within the limits where you believe you need to be for regulatory acceptance. And you want to do that with a reasonable effort. And of course, it's expected that your assays will get more precise over time, and it's assumed that the accuracy won't change. So here's a second version of the assay where the noise is reduced and confidence intervals in green there. And then a third assay where you've actually got your confidence intervals within your target specification range. Again, this is not a robust statistical analysis, but I think that I want to illustrate a point here. I'll point out here, even with a good assay, an assay with a relatively low percent CV, even a sample that's actually at 79%, let's say it's truly at 79%, you know that. You can make a sample that has a concentration of 79% of your target, test it, confirm yourself with this, with the assay noise that you have. You're still going to pass a limit if it's right there, almost half the time. So this is the dilemma we have. We don't want to pass a sample that's really subpotent, but we don't want to fail a sample that's good. That's our big challenge. So you could just tighten the spec range, and then you're limiting that type 2 error of passing a bad sample, but then you're increasing your risk of failing a good sample, a perfect sample actually, a type 1 error. So balancing these factors and being able to understand that and explain that not only to health authorities but within your own company is critical. And of course your statisticians will help you determine how many replicates you need to achieve a target specification, but nobody wants to run 20 plates or 50 plates to get one result. That's not going to be popular. So again, it goes back to taming the noise in your assay as early as possible in development does pay benefits down the road, even though most of the drugs you're supporting are not going to make it. You need to advocate for what you're doing. So as you know, the specification range may be determined by the inherent noise. The goal is largely binary, passing a good batch, failing a bad one. I tried once to, within my company and one of our filings, to not actually report a number because it's so hard to explain. People always see the number even if it's in with the noise of the assay and they think there's something going on. So I just said, well, it passes and the spec is this and this. And people were mildly insulted by that. In fact, the FDA person said, I think I can understand this data. I'm not sure they could, but I gave it to them. I didn't try that tactic ever again. But at some point, there's no improvement that can be made. So there you're stuck with the performance of your assay. And then you have to deal with that on the spec. And when your assay noise is too high to where you're not able to meet some regulatory request, that's when you really have to share a lot of data and convince them that this is as good as it can be. And I'm sorry, but that's the nature of this test. But you need to be competitive. So if your assay noise is here and most of what your health authorities are seeing is here, you're going to continue to get challenged. So that's why it's great to come to these kind of conferences and hear talks, because you kind of know where you're at and where you need to go. So I'm a big advocate for that. I think I've covered this. And we talked a lot about this yesterday in the workshops, but monitoring, there's so many important aspects of the bioassay, be it the asymptotes, the R squared, the goodness of fit, the precision. There's so many parameters that can be monitored, and so much easier to do that today with the modern digital tools that we have, that using that to set up a robust assay acceptance criteria, which must be satisfied before the sample is actually even analyzed, is the best way to prevent creating concerns and potential out-of-spec investigations that will put you in the headlights and delay you and cause all sorts of burden of explaining why a retest is needed on the product. That's the worst possible scenario. What's wrong with your assay? And maybe there's nothing wrong with it, but you want to exclude an assay before you start down that path, particularly as you're later in development or your commercial lab is having issues with the method that you've developed. And of course, degradation and stability studies are gold mines to monitor bioassay performance. Sometimes your molecule is just incredibly stable and you can beat on it, and you don't want to. You want to treat it in a way that's relevant. I think Scott mentioned yesterday, it makes no sense to boil your sample and get no signal and say, look, I have demonstrated my bioassays stability indicating. So this can be a fair bit of work. So what I would recommend is you really utilize your stability studies that you have even before you go into forced egg studies. And when you go into forced egg, think carefully about how you're going to treat the molecule to where you can get gradations of potency in a more meaningful way, as opposed to the very harsh treatments, which is not going to win the argument anyway. So formal stabilities typically include, pH shouldn't be in there, but temperature and light. So you've got that information. So here's again a case. I think this will look familiar to it. A case where your specification might be 80 to 120%. Assay 3, a tight data. This is using the same data, but ordering it a little differently. Sometimes you see this phenomenon, what looks like something's going down, there's a trending down. And there becomes this great concern within the company that the next time point's going to go out and you're going to have a clinical supply issue. So what do you do in this scenario? Well, you could wait for the next time point and stress for 3 months or 6 months. Or you can have retained samples and do an additional test. And that's often helpful. And that's perfectly allowed as long as you have the material to test. So plan for that. Because sometimes it pops right up and then you're not worried anymore. And then you look at the data in toto, this is an exaggerated scale. If I put that on this other scale, this data would look flat. It's hard to explain this to folks sometimes, so I think this is one of the tricks you can use to help keep everyone calmed down in these scenarios where data coincidentally is trending a certain way. Yeah, this is that same data in a different scale. So my advice would be to understand your method variability before you start a stability trial, or you'll run into these problems. And of course, there's many other parameters, as we've mentioned, other than the numbers. So we look at a lot of other things. And this is from a recent publication. Curve shape. I won't go through this because I know everyone's very familiar with this, but I just want to point out the assurance that the molecule is behaving the way it should is not just based on the number, and it's not just talking about the noise in that number. So I'll change gears in the last 10 minutes here and talk a little bit, it's a big picture items. No one needs a reference standard more than the bioassay lab. And you need to push for this to make sure it gets done and it gets done properly. The material is made in sufficient quantity, stored properly. Otherwise, you're going to have a problem. You need to confirm that it's not going to drift over the storage time, and that requires collaboration with the other units in your organization. Again, making the largest possible amount is always a good idea, because switching is going to be difficult. It's going to be laborious. You want to track your reference standard performance every time it's used. Look for that drift. Look for the little incremental changes that may be occurring in the curve shape. Replacement lots. This is always going to be a challenge for you because there's a regulatory opinion on it. There's internal opinion on it. And if there really is a change in potency from one reference material to another, you've got a large burden to manage that for the life of the program and explain that for future generations. So it's always easier to claim the materials are equivalent and design that with a high replicate study. I think we talked about this quite a bit yesterday. If you can confirm these two materials are not different and not have to worry about adjustments in potency, it's always going to be easier. But you need a number of lots to be able to choose an appropriate one for that. Okay, I think I've covered this. I'll just point out, one of the frustrating things is you have to satisfy all the regulatory agencies in the world, and they sometimes want different things. Even within a country, you can see differences in expectations. And if you're filing in both, you have to be aware of what's going on. Again, that's why meetings like this are great. I think the FDA folks couldn't come today, unfortunately. But I know we have European regulators and others here, and it's so helpful to understand and talk face to face. When you don't understand why you're getting a question, to be able to meet them around coffee and talk through these things, it's very important. That's how I got probably some of the best information I have and was able to interpret some of the feedback we were getting formally. All right. Successful strategies for bioassay. Again, be aware of where this drug is going to be filed because they may have special requirements that you need to meet. Sometimes I've seen folks that have some rather bizarre ways of improving their assays, like putting plates in a drawer. Odd things, People are people. They sometimes want to believe, even scientists sometimes want to believe in magic. Try to avoid that. If they have a hypothesis, let's test it scientifically. Again, start as early as you can. And, we talked about the development mode of a drunken sailor approach yesterday as opposed to a defined pathway. Sometimes the drunken sailor approach works, but most of the time it doesn't. So, having a team of folks to help guide you through the best experiments first can really save a lot of time. And that requires not just being methodical, but also, again, having the right people there. Sometimes it's good to bring a person outside of the bioassay group in, fresh set of eyes can really be helpful. Sometimes our statistician will help with this. He'll ask the right questions that we won't ask ourselves because we're just too close to it. On stability, if you do get a weird result, having a retest procedure already written, so you're not just trying to give the appearance of testing something in compliance can be a great help. Like if there is something that's off, I mentioned you could test another sample or you could test that same sample again if you have a solid scientific argument for that. I'm always a little leery of outliers, even though, of course, if you have 3 reps on a plate, you should be able to throw one out. There's plenty of tests to do that. It seems perfectly valid. It's just always difficult to explain because people still go back to that, I think it was the 80s when they had the Barr decision. Company was, testing material into compliance by testing it repeatedly and applying, perhaps an imperfect way of removing outliers. If you're going to do outlier testing, it should be done on your data all the time, not only when you saw a result that looks odd. That's my advice on that, to avoid a lot of questions, not only with health authorities, but also within your quality units and your company. It can be a hot button issue. I've already talked about having a close relationship with a dedicated statistician, and by that I mean someone that really understands the science of what you're doing, and they're going to have to invest the time to do that. It can't be a part-time job where they're coming in at the end, as I mentioned. Again, another thing is just be aware of where you're going to run your bioassay for what seems like a long time, but it's actually a short period of time in the lifetime of a product. Another lab's going to be running it. They may not have the material that you have. So if you can anticipate where it's going, be it a CDMO or your own commercial lab, and you know what they may have country requirements where they literally can't buy and use the material that you use in your assay. So the more of that you can anticipate, the better off you will be. And I already mentioned this issue with assay acceptance failure rates, being perceived as a problem to be solved when it was actually a design feature. I already mentioned that, but I'll say it again because it's something that surprised me a little bit. And having a priori procedures for handling inspections is good. I've already mentioned that. Sometimes it's just a noisy assay, and the best people in the world cannot get it any better than it's going to be. And there's your inherent noise in the assay. It's the behavior of the cells. Maybe you're working with some difficult cells. One approach is just to simply increase the number of tests. So, again, going back to the case that I gave you, the moving average on this is 99.3%, around 100. But if you only tested this once, you can see that you're going to be outside of your specification range. Again, this graph here is a moving average. So you can see that if you tested it 5 times, it gets to 101%. So that's 5 plates. And then it doesn't really help if you test it 20 times or 5 times. So sometimes you can just get over assay noise by increasing the number of plates you run. Of course, this is more costly. Time affects your assay throughput and turnaround, but statistical approach can help you get this done. Okay, my last 5 minutes, and I will take a question or two if there's time. I guess my main advice, looking back on this and having led a group for a decade, is you need to be an advocate for what you're doing. Explain the value of what you're doing, why it's necessary, and how to interpret the data. We talked about this yesterday as well, understanding when it's a QC test and what features of the drug are a QC test and which can be a characterization test with a lower bar will always pay dividends. I'm really repeating myself here. Control samples can be helpful. How will you handle trends? We talked about binding assays. It's common on the relative potency assay to start phase 1 with a binding assay and then transition to a cell-based assay. But it's also quite possible to transition back to a cell-based, an ELISA. But you have to have the data to support that. You have to show that your bioassay is not inferior in any way. Your binding assay is not inferior in any way to your potency assay. It can actually be much more precise, of course. And in some cases, there are no availabilities of cell-based assays. I think we had one experience where the discovery unit was isolating neurons from the fetuses of mice and exposing them to the drug, and that was the cell-based assays. Now that's a primary cell. And it's really bizarre. We were able to show binding assays far superior, and that would have carried us all the way through to commercialization. Sometimes there's just no way to put your target ligand into a cell that's viable for a cell-based assay. That can happen, it's rare. Okay, my final tip is the bioassay scientists present to your group or in conferences as often as you can. And particularly when you're speaking outside of your area of expertise, try to avoid jargon, acronyms. Try to make sure people are really following what you're doing, because people will nod their head, but most of them don't know what you're doing. They just don't. It's very complicated. And most folks don't understand statistics very well. They don't understand that variation. And they get angry when you tell them that your tool of measurement has more noise than the production controls and when they're making the drug. And that can happen from time to time. But it's still necessary to do the test. We probably overdo it, but that's the world we live in. and we have to satisfy all regulatory expectations. Okay, I won't go into the bonus slide, but I talk a little bit about, if you want to look at these later, I talk a little bit about bridging assay, in this case a binding assay and a cell-based assay. And it's pretty easy to tell if you're using, but you really have to be careful how you do your degradation, that you do in a relevant degradation, a mild degradation. Sometimes you can do very limited proteolysis. Sometimes it's a gentle heating, sometimes it's a slight change in pH. You want to get that curve moving on the relative potency assay. The other thing you can do is look at your stability data. If you're running a cell-based assay, you can just take, when that samples start at accelerated conditions, start moving off 100%, you can take those samples and run those. And if you can also see them in your alternate assay, your binding assay, you're building a case for a bridging an assay. So sometimes that's a convenient, less costly approach. Okay, I'm at time. I might be able to take one question, but I'll be around the rest of the week. So I really appreciate the invitation. I'm looking forward to hearing from the rest of you.
Thank you, Ned. Thank you, Ned. We have time to take a couple of questions, but really nice job in highlighting sort of the ecosystem, right, of what we have to deal with in ensuring that we've got an assay under control. And so we'll open up for a couple of questions.
Okay, well, I can yell. Thanks, Ned. Great talk. Couple of quick questions. One, could you talk a little bit about what you think is the right time to establish a cell bank for a cell line that you're using for a bioassay, when in development would be the ideal time to do that?
I would just say, as soon as you start developing the cell-based assay and you begin to understand enough to where you're reasonably confident that will carry you through to the end as soon as possible.
Sometimes not in phase one.
Yeah, typically not.
A little later than phase one.
Yeah, of course it depends on your time. Nowadays, we're trying to push everyone to go so fast. You almost feel like, you just take a breath from phase one, it's time to get going. So you can overinvest can probably do it too early, but you don't want to be caught short either.
Okay, and second quick question. Occasionally you run into a bioassay where you try to do stress stability and you hope that you're going to see a gradual decline, but occasionally you run into one where there's no effect, and then it drops off a cliff. It's almost like a switch on and off. So how do you deal with that type of thing?
Yeah, I talked a little bit. I think we should be a little more creative and think, maybe not rely just on ourselves in the bioassay lab, but go to the rest of the analytical organization and say, look, I want to gently degrade this molecule, and you can do some things like an extremely low level of a protease, for example, or a very low temperature, which can sometimes just take too long. And then you've got the protease in your sample. That's a problem, right? So you could put the protease on a bead. Actually, I'm not sure we ever did this. Dave, we talked about this. It's an idea I had, but to degrade something to where it actually begins to lose activity, but it's not just dropping off the cliff. My advice would be to engage others. There's always smart people around, and if you present the problem to them, you can get help with it. I don't have a simple answer, though. Okay, thank you.
It was a great talk. You mentioned something about building a 5% failure rate in a commercial assay, not building, but having the 5% failure rate, and then explaining to people why that was needed. Can you elaborate on that a little bit more?
Again, the principle is you'd rather fail an assay than a product. or make a product suspect of being out of spec, because that's the worst possible scenario for the company. I mean, passing a bad sample is the worst possible scenario, but you've already accounted for that in your spec. So your risk is that you're going to fail a good sample. So you can tighten your requirements for your reference standard performance or your control sample as tight as you want. So you could get a failure rate of 50%, fail 100% if you made it too tight. You got to think about, where do you want to be? What can you live with? Can you live with failing a perfectly good assay 1 in 20 times? That seemed like a reasonable compromise. That's how I got to 5%. Your company might say it should be 10%. But you got to find that sweet spot of you want to fail an assay that's not perfect so that you're not putting the product at risk. But as I said, Commercial Labs considers that design feature actually a flaw and something they continually work on. I'm like, you're not going to fix that unless you loosen the criteria. And then you run the risk of passing an inferior product. That's a difficult conversation. That's why I was saying, have that conversation early, decide what you want to do and sell it.
Any other questions? Go ahead, and please identify yourself and the affiliation, your affiliation.
Yeah, sure. I'm Long from Bristmeyer. So I have a question about the example you mentioned about neuron cell, that one. So what you have done to convince the health authority to accept a binding assay instead of bioassay for the filing?
Yeah, I was just saying, if there's no relevant, meaningful cell line available, and the best you can do is a binding assay, you can convince a health authority that is appropriate as a relative potency assay.
So you will need to provide a data package to demonstrate that?
Yeah.
Okay.
Show all the data where you tried to make this assay. And of course, you can appeal to things like, we want to avoid unnecessary animal use. So clearly that was the case here for us. That's part of the argument, but the main argument is it does not provide any additional benefit from other than what you would get from a binding assay.
Was that a long debate, or it's like you present?
No, it was a lot of work, but it was not a long debate. It was obvious when we were done what the best solution was. It was not a hard sell to health authority.
Okay, thank you.
I think it doesn't work. There is a guidance on how to set potency of reference standards. He talked about reference standards and said, do as many vials as you can. The guidance says the potency is to set 100% if the values range between 90 and 110%. We were really struggling with this because the question is, if you have to do the next reference standard, how do you manage if you have some manufacturing variability or other sources of variability if you always set it 100%, which represents the assay variability, I would say. So we were struggling with this definition. It's still a draft guidance, but maybe, you have some ideas on this.
There's been entire conferences on this subject. It's a very challenging problem. The argument is always, well, if it's really 10% off and you're doing multiple requalifications of multiple reference materials down the road, you're going to see this drift over time, and pretty soon you're going to start seeing a bias in your data. But in reality, that's not common. It may be in some scenarios, and you're right, maybe 90 to 110 is too loose. But if to just automatically reset it at 100, that's a good debate to be had. Mark, you got an opinion on this?
Yeah. There's a couple strategies you could use. if you have multiple manufacturing lots that you can select from, you can do extra replicates on each of those lots and try to find a batch which is more centered, closer statistically to the previous reference standard. So also for when you're qualifying a new reference standard, you need to have an SOP that usually includes a tighter acceptance criteria for the new reference standard than the actual lot release spec. So those are two things that can help.
Great questions, everybody. Let's thank Ned again for really starting the conversation for the rest of the session.